American, almost every issue had a big breakthrough in biology. So if you look at the 1970s in biology, it was the first time people were able to sequence DNA, so directly obtaining genetic information, in the language of the gene. And then people were able to solve the first structures of big biological molecules. They were able to understand what the cell membrane looked like, how cell membranes worked, how the immune system worked, a lot of breakthroughs in neurobiology. So it was a very exciting time. And I have to say, that revolution in biology is still continuing. So we're still at a stage where biology is undergoing a huge revolution. And in fact, if I have to say the two big areas where there's a revolution going on, it would be modern molecular biology and artificial intelligence. And actually those two are coming together. So then I realized that actually many physicists had made a transition from physics to biology. So Francis Crick is one of the most famous of them, but also Max Perutz, who founded my institute, and Max Delbruck. So all these people had started off as physicists and then gone on to do good work in biology. So I thought maybe, I could do that too. And I thought of doing a postdoc in biology, but then I thought if I did a postdoc, I would get a very narrow education, because I would be trained in just this narrow area where I was doing a postdoc, but I wouldn't get a broad background in biology, which I think is useful. So I realized I needed a basic background because I didn't know any biology. So I decided to go to graduate school all over again. And in fact, I applied to lots of graduate schools, and one of them, Yale, said, we're not going to accept you as a graduate student because you already have a PhD in physics. But I'm going to send you a CV, and maybe some faculty members will want to hire you as a postdoc. And I got two letters from two faculty members at Yale. One of them was Tom Stites, with whom I ended up sharing the Nobel Prize, But I told both of them, I'm sorry, thank you very much, but I don't know any biology, and I'm not ready for a postdoc. So by this time I was married, and I had a stepdaughter who was age 6, and then I had a son who was only about 6 weeks old. And so we had to pack up all our stuff in a Ryder truck and drive from Ohio to San Diego across the US. So then, I spent 2 years learning biology here at the University of California in San Diego. It's a very beautiful place right on the ocean, and very well known for biology, and lots of institutes around the university which also specialize in biology, like the Salk Institute and so on. And this is when, after a year and a half, and it's very funny, when I started my first week there, the first week for new graduate students is a bunch of lectures given by all the faculty about different areas of research, about their area of research. And I didn't understand a thing they said. For example, there was a professor I remember who kept talking about lambda. To me, as a physicist, lambda meant a wavelength, some other kind of constant, And but to a biologist, lambda is a bacteriophage. It's a virus that attacks bacteria. So I realized I had to take undergraduate courses. I was not good enough to even be a graduate student. So I went to my advisor and said, look, I need to start at the beginning. So I want to take all these undergraduate courses. And luckily, he said, fine, you should do that. So I'd taken all these courses, I'd done a lot of lab projects. And then in my second year, I thought, well, actually, I now have a background, and I can do lab work, I know how to work in the lab, and so on. And at that time, there was an article in Scientific American written by two professors at Yale on how to apply a physical technique called small angle neutron scattering to study the ribosome. And it turned out one of the authors was one of the professors at Yale who had written to me asking me about a postdoc. And by then, of course, I knew that a ribosome was really important. It was a central molecule in the cell. And so I said to him, look, I wrote to you, you wrote to me and expressed some interest in me when I knew no biology. And now I actually know some biology, so maybe you're still interested, and if you are, I'd like to consider a postdoc. And he said, well, we'd like to have you as a postdoc. Why don't I put you in touch with my collaborator who works on the ribosome, Peter Moore. And so I ended up accepting a job, a postdoctoral offer with Peter Moore at Yale University. So after only 2 years, again with my young family, small children and my wife, we took off to go to the other side of the country, to Yale University in New Haven. And one of the lessons as I look back in life is, if you're going to do science, you need a supportive partner, because my wife was very agreeable, saying, okay, we'll decide it. And this was really a lot of my career is because she was willing to adapt to what I was doing. And of course these days it's much harder. My wife is a children's book writer and illustrator, so she can work from home. But with two sort of real, official careers where you have to go to work in an office, it becomes even harder. So this is Peter Moore, my professor at Yale, my postdoctoral mentor at Yale. Around the time I was in his lab, and he was a student of Jim Watson. And today we know about Jim Watson mainly for DNA and for making really stupid remarks to the press about gender and race and so on. But Jim Watson's lab also was one of the labs that discovered mRNA. And a lot of the early work on ribosomes came out of his lab. And many of the leaders in the ribosome field came out of Watson's lab. And in his lab, we were trying to understand where the different proteins in the small subunit were located by replacing each of these proteins with a deuterated version of them, and then doing small angle neutron scattering. And deuterium has a different scattering factor for neutrons than protons do, so you could locate where all these proteins were. But of course, just locating them, without any other fine structure, doesn't actually tell you how a ribosome works. And I like to give you the analogy of a car. If you look at a car like this, you're not going to be able to tell how a car actually works. But if you start looking at how the car is sort of organized, you get a better idea of how it works, but you still don't understand how it works. It's only when you see all of the details of the car that you can have some idea that, oh, this is an engine, it has pistons move and they compress, and the force of compression can turn the wheel through a crankshaft and so on. So you can understand how this works. And it's the same with biological molecules, or actually any chemicals. And this leads to a question of how you visualize small objects. So the ribosome is very small. How would you see details in a small object? And the typical way you do it is, you would magnify a small object with a lens, which would project a magnified image, and then you can look at the magnified image and see details. And effectively, what a lens is doing is collecting all of the scattered rays from the object and recombining them into a magnified image. And of course, if you've studied physics, you know that what a lens effectively is doing is doing a Fourier transform on the scattered rays. And the trouble with molecules is, if you want to get molecular structure, you cannot use light microscopes. And the reason is that light has a wavelength of 500 nanometers. And of course, there's an old theorem from optics, which is the Abbe limit, named after Abbe, the German physicist. And the theorem basically says you cannot resolve anything that's closer than half the wavelength of the radiation you're using. And the distance between atoms, which is known for a long time, is about 0.1 nanometers. And so you're 2 or 3 orders of magnitude off from where you want to be. However, there are other kinds of radiation, most notably X-rays, which are essentially the same particle photons, but they have a wavelength that's comparable to interatomic distances. But the problem is X-ray microscopes. There are X-ray microscopes, but they're only suitable for looking at objects of a cellular level. They don't have the resolution to look at molecular structure. And the other thing is that if you use enough X-rays to look at a molecular structure, you destroy the molecule before you got enough signal. So the way around that is a way that was discovered first for any molecules in 1912 by von Laureate and Lawrence Bragg and others, is to crystallize molecules and then hit them with X-rays. And because of the regular arrangement, the scattered rays only go in certain directions, which are these X-ray spots or diffraction spots. And you can measure those scattered rays. And if you can measure them, then maybe you can do it in a computer what a lens actually does, which is you can calculate the Fourier transform of the scattered rays and end up with a 3-dimensional image of the object. And that's how crystallography works. There are a lot of details that I've skipped over. But of course, I didn't know any crystallography. And by this time, I was a group leader at Brookhaven National Lab in the eastern US on Long Island. And so when I got tenure, I was allowed to go on sabbatical. So the first thing I did was to go to Cambridge in England to the MRC Lab of Molecular Biology, and that's the building as it looked then. And I wrote to Aaron Klug, who was the director, and said, I would like to come and spend a year sabbatical in your institute to learn crystallography. And luckily for me, because I was not at all well known at the time, he didn't throw my letter into the waste paper basket. Rather, he replied to me and said, yes, we're interested. And he sponsored me for a Guggenheim Fellowship, and I was able to go to Cambridge. And so I learned crystallography. I came back to Brookhaven, but I was not really happy with the way that DOE was funding national labs at the time. So I moved to the University of Utah, which had a very strong program in biology and genetics and RNA especially. And when I went to Utah, and actually even slightly before, when I came back from Cambridge, I had the idea that you ought to be able to use crystallography to tackle the entire ribosome. That too has a history. So in the 60s, people had shown in the 60s and 70s that ribosomes in certain tissues would form 2-dimensional arrays, very regular 2-dimensional arrays. And this was the first evidence that ribosomes were probably identical particles. Before that, they weren't sure, is every ribosome the same as every other ribosome? But actually, the fact that they could crystallize shows that they were, close to identical. And this is, but there were no 3D crystals until Ada Jonat went to the Max Planck Institute, run by Heinz Gunther Wittmann. And Wittmann was very interested in ribosomes. His entire department worked on ribosomes. And together they published the first paper on true 3-dimensional crystals of ribosomes. And a few years later, a group in Russia, headed by Marina Garber here, and this guy, Murat Yusupov, you should remember him because he shows up later. So Marina Garber's group obtained crystals of the small subunit and also the entire ribosome. So now we had crystals of both the small subunit, the large subunit, and the entire ribosome. Now, initially, these crystals didn't diffract very well. That means the crystals were not of good enough quality to get an atomic structure. The order inside the crystals was not good enough. to get an atomic structure. But gradually, Ada was able to improve the large subunit. So in the mid-1990s, there were 15 years had gone by after the first crystals, but there was still no structure, not even a low-resolution structure. So you should have had a low-resolution structure by then, but there wasn't. And Jonas had produced improved crystals of the large subunit, but still several years later, there was no credible map, low-resolution electron density map to show that some progress was being made. And so, after a couple of meetings in the mid-90s, the field realized, or at least some of them came to the conclusion, that Jonas' group, which was very well supported, was basically stalled, and that maybe the field needed new people with different ideas to come and work on it. And the Yale group decided to take the 50S crystals that Ada Jonas' group had crystallized and decided to see if they could solve it. And I thought, great, because those were the best crystals. And I thought, let Jonas and the Yale group, they can fight it out. And I thought there were no good crystals of the small subunit or the whole ribosome. But I thought, well, maybe the whole ribosome was a little too difficult to try to improve the crystals. But maybe I could tackle the small subunit and try to improve the crystals to the point where you could get an atomic structure and then try to figure out how to solve it. So I was at the University of Utah at the time, and I was joined by, this was my group at the time. And if you notice, I like to joke that I'm in my Bin Laden days. But I'm also wearing gloves. And this shows you I was the only guy doing any experiments with this picture was taken. Okay. But this is Bill Clemens, who's now a faculty member at Caltech, and this is John McCutcheon, who is now Howard Hughes investigator at Arizona. And this is Brian Wimberly, who's at the New York Structural Biology Center. So anyway, these two guys were first-year graduate students, and because they didn't know anything about the ribosome or crystallography, both of them said, yes, it's a good PhD project to try and solve the 30th subunit. And this shows you, if you don't know something, then you don't know that it's too difficult. And so you decide to go for it. So sometimes it's a disadvantage to know too much. This is Brian Wimberly, who was very knowledgeable, and he was a little cautious initially, but then he joined the project a little bit later and played a very important role. So we started with the small subunit, and to our surprise, we very quickly, within a few months, were able to obtain much better crystals than what had been published, more than 10 years ago, 10 years earlier, okay? So this shows you that if you pay careful attention to biochemistry and the quality of your sample, then you can sometimes make an improvement. Now, crystals of the large subunit have been around for many years, and still there's no structure. And so the question was, what is the difficulty? And the difficulty is that something to do with, in crystallography, what's called the phase problem, which I won't go into. But one of the ways that protein structures were being solved was, if you look at this mirror symmetry, many crystals will have mirror symmetry. In fact, all crystals will have, symmetry of this type, where an X-ray reflection will have a symmetry-related reflection on the other side. And normally these two reflections are very equal in intensity. But if you have atoms which have a special absorption property, where you're exciting a state and then there's a re-emission, then what that does is it creates effectively a scattering property of these atoms called anomalous scattering. And that gives rise to a small difference between the left and right half of these images. And that left and right difference can help you solve the phase problem. The phase problem is that, when you measure these spots, you're only measuring the amplitude of the X-ray wave. But you need to know where the peak of each wave is relative to all the other spots. Okay, so these different spots will lag behind or be ahead of each other. And so that problem is called the phase problem, and that's the big problem in crystallography, because you can readily measure the intensity, but to get the phase you have to use other procedures. But anyway, this idea of using selenium was touted by people like Wayne Hendrickson. But it turns out there are stronger anomalous scatterers. And there, for example, the L3 edge has like 15 or 20 electrons of scattering power. But one of the compounds is called osmium hexamine, and its L3 edge also has about 15 to 20 electrons. And when I was starting this project, I read a paper by Jamie Kate and Jennifer Doudna. Of course, Jennifer Doudna is now very famous for CRISPR-Cas, but for a long time in her life, she was an RNA structural biologist, sort of like me, and before she went off to, fame and glory. So they had solved this complicated RNA structure using osmium hexamine. So we decided to use that. Now, while all this was going on, what had happened is that the Yale group started making progress on the 50S. So Ada Yonak switched her attention from the original 50S to the 30S, and also obtained some reasonably good crystals, which were not quite as good as ours, and so her structure in the end didn't turn out to be as complete or accurate. But she was working on the 30S, so suddenly my two graduate students were now competing with a big group that had been working on ribosomes for 15 years. And then the Yale group had assembled a very strong team. This is Peter Moore, my postdoctoral mentor, and Tom Stites. And these are their two postdocs, Nana Ban and Paul Nissen. So, outstanding team at Yale. And then Harry Noller, who's a very famous ribosome biochemist, recruited that guy, Murat Yusupov, whom I showed you, and his wife, Gulnara, from Russia, because these were people who had crystallized the original 70S. And then he also recruited Jamie Kate, who had developed osmium hexamine, who had first used osmium hexamine. So this was also another strong team. So suddenly, what I thought would be a quiet life working on the 30S became like this four-way race. Okay? Now, if you're in the middle of an intense four-way race, what you should not do is move. You should stay where you are and focus and work hard and try and get there before everybody else. Because if you move, it will cost you a lot of time. Okay, and so the thing I did was right in the middle of the race. I moved not only to a different university but to a different continent. Okay, I moved from Utah to Cambridge, to the same institute where I had done a sabbatical many years earlier. So you might ask, this is a crazy thing to do, and why would I do that? Well, I told you I wanted to work on whole ribosome structure, and I had no idea how long it would take, because even though there were crystals, there had been no structure for many years, so I didn't know how long it would take to solve this problem. I had these ideas about using anomalous scattering, but I didn't know if it would work. Okay, so I thought, well, if I stay in a university you have 3 to 5 year grants, typically from the NIH. And if at the end of the third or fourth year, if you don't have a big breakthrough and publish, they're not going to renew your grant. They're not going to say, oh yeah, keep doing what you're doing. So that's a problem with that sort of funding. And then I thought I would need expert crystallographic colleagues in case of serious technical problems. I would get help. And the MRC lab is one of the places where the methodology was developed. So they had people who really understood all the software, data collection and so on. So I thought, if you're pushing the technique to the limits, you might as well have all the experts in that technique nearby so that you can consult them. And finally, and also it's a collegial atmosphere. It's, the groups at the MRC are very small. They're about, typically 8 is a very large number. It's like the upper limit. And most groups are about 5 to 6 people. And what this means is you're forced to focus on the problems that you think are the most interesting, because you cannot do everything. So if you can't do everything, you really focus on, you have to pick and choose which problem you want to work on. And I think that's one of the reasons, this combination of stable funding and small groups is one of the reasons why the MRC has done so well. Because this lab alone has produced about 17 Nobel laureates. And it's not that big an institute, compared to big universities like Harvard or something like that. And then it has a long tradition of tackling important problems. And the emphasis is always on the importance and quality of your work, not on the number of papers you publish. For example, Fred Sanger, has an H index of 18 and, about 40 papers or so, and won 2 Nobel Prizes, okay? And so that's an example of the sort of thing that they like to do. So I persuaded my wife that I should take an almost 50% salary cut and move to England, from Utah. And she said, we had spent a year sabbatical, so she said, okay, I'll move, but this is the last time I'm moving. So, so we left. Our children were still in the US, they had grown up by this time. And so we left our children and went off to England to pursue this ribosome problem. And my wife has kept her promise, because every time I get a job offer back in the US, she said, no, we're not moving. I already told you this is the last time. So I had to also assemble a team in the US. And luckily, 2 of the key people from Utah agreed to come with me to England. But then others joined, so 2 of them, so Andrew Carter and Ditlev Brodersen joined, and James Ogle came about a year later. And these were the people who worked on the small subunit. And of course, for these very large molecules, which produce very small crystals, the diffraction is very weak. And to get a strong signal, you need these very powerful X-ray sources, which are synchrotrons. So I like to joke that if you could join a ribosome lab and see the world, because these are all places where we collected data. This is Long Island and France and Chicago, but they're not going skiing in Grenoble or listening to jazz in Chicago. Most of the time, they're only seeing the world's synchrotrons, which is to say that they're seeing the inside of a diffraction instrument and a beamline in the synchrotron. And this is the X-ray beam which hits the crystal, and the crystal is then, the diffraction from the crystal is measured on this detector. So using this, we were able to solve the entire structure of the 30th subunit at about the same time that the Yale group solved the structure of the large subunit. And one thing that came out of both structures is that if you look at this pocket, this is the pocket where the amino acids are joined in the large subunit. And if you look at this little cleft between what's called the head and the body, there's an area around the neck. That's where the mRNA binds and where the tRNAs recognize the code. So those are the functional regions of the ribosome. The functional regions are all RNA. There's almost no protein where the functional regions are. The proteins are all mostly on the outside of the ribosome. And this tells you something, because when the ribosome was discovered, there was a big puzzle, which is the ribosome is the molecular complex that makes proteins. But the ribosome itself is made of proteins. So how could the ribosome have got started? Because where did the first proteins come from that made the ribosome? And it was actually Francis Crick who first had the idea that maybe early ribosomes were entirely made of RNA. At the time, there was no feeling that RNA could be catalytic. They thought RNA just carried genetic information. But in the 80s, Tom Check and Sidney Altman discovered RNA catalysis. And so they realized that RNA could both carry genetic information, like DNA, and also carry out chemical reactions, like proteins. A single molecule could do both. And so this led to what's called the RNA world hypothesis, which is that life began where RNA was both the information carrier and the catalyst. And then eventually, it slowly morphed into today's world. And today's ribosome became more complex with proteins added on. And a number of biochemists, notably Harry Noller, produced a lot of biochemical evidence to show that. So here's our complete atomic structure, and as you can see, there are 21 different proteins, actually 20 in the structure, and the very large RNA molecules in different domains. Now, right when we solved the structure, we could immediately solve the structure of the ribosome bound to antibiotics. So many antibiotics, possibly almost half of known antibiotics, target the protein synthesis machinery. Many of them bind to the ribosome. And so we looked at many different classes of antibiotics bound to the ribosome. And that, once you have the basic structure, it's very easy to solve the structure with the antibiotic added. And so these were some of the first antibiotic structures bound to the ribosome that came out. And then a few years later, we ended up solving the structure of the entire ribosome, and that's thanks to another group of people. And this structure, which came out in 2006, was actually one of the largest structures solved at the time. And the whole ribosome from a higher organism contains about 500,000 atoms. This one contains probably about 300,000 atoms in the structure. But actually, there were two molecules in the asymmetric cell, and in the unit cell, two molecules in the asymmetric unit. And so we had to solve sort of a 600,000 atom structure, to do this. Now, one of the important functional things that came out of our work is how the ribosome ensures the accuracy of translation. That is, how does it translate the genetic code so accurately? Its error rate is only about 1 in 10,000, 1 in 1,000 to 1 in 10,000. Now, here's the genetic code. And if you look at it, you can see that often many three-letter units will code for the same amino acid, and they only differ at the third position, at the last position. And this led to the idea that the same tRNA can recognize different codons, even though at the last position it's not an exact match. Okay, that's called the wobble hypothesis. Now, tRNA selection is also very fast. So here's a ribosome waiting for a new tRNA. And like I said, its accuracy is slightly a matter of dispute, but at least 1 in 1,000, and some people think it's much higher. And so it has to choose, in the cell there are many different tRNAs, and from that it has to choose only the tRNA that corresponds to the three-letter code that's in this site where that tRNA binds. So this is a problem that was solved in many steps, but I'll just show you the final structure that we ended up doing, and that's the work of Martin Schwing and Rebecca Voorhees and Kelly. And Kelly is now retired. Rebecca is a faculty member at Caltech, and Martin is at McGill University, Canada. And so this is the structure that they ended up solving. So they basically figured out how to trap tRNA with this protein that brings the tRNA, that delivers the tRNA to the ribosome. Now what happens is, if the tRNA matches, then it sends a series of signals to EFTU, which is this protein. And this molecule, EFTU, then hydrolyzes GTP. When it hydrolyzes GTP, it changes its shape and allows the tRNA to enter the active site where the peptide bond can be formed. And so I just want to show you this in a series of events in a movie. So here's a ribosome waiting for the new tRNA. And the new tRNA is brought by EFTU. And now this tRNA can actually have a variety of conformations, including, see, this bent conformation. The bent conformation is normally unstable, but if it matches the codon, then the base pairing and changes in the ribosome stabilize that bent conformation. And it's partly stabilized by these bases from the ribosome itself. So the ribosomal RNA changes conformation. And what these bases do is they recognize whether there's a Watson-Crick base pair at those first two positions. That's how they check whether the tRNA is correct or not. And if it's correct, that whole conformation is stabilized, but then parts of the ribosome move towards this factor. So you can see that what happens is, this is a part of EFTU, and this is a part of the ribosome small subunit, and they change conformation, so they stabilize this complex, and this end of the tRNA also changes its conformation. Now, when that happens, one of the other things that happens is that there's this histidine, and there's a histidine in EFTU. This is the gamma phosphate of GTP. This is the final phosphate. And the histidine changes conformation because it's stabilized by a loop here, the Laureate subunit, and results in hydrolyzing that phosphate. And when you have a GDP form and the phosphate leaves, the whole EFTU changes its conformation. And when it changes its conformation, then what happens is that it loses contacts with the rest of the ribosome. And this tRNA then swings into the peptidyl transferase center, and then an amino acid is added to the growing protein chain. So this is a long, complicated series of events, and it took us, probably about 10 years from starting the work to the point where we could get the final structure that allowed us to do this. And of course, over the next 10 or 15 years from the first structure, we ended up trapping the ribosome in many different states, and then trying to get crystals of each of those states, and basically getting snapshots of the ribosome in action as it did it. But that's a very difficult problem, and the states kept getting harder and harder to trap, kept getting harder and harder to crystallize. But luckily, there was a different technique that suddenly became more powerful. The technique itself is quite old. It was electron microscopy, and this is Ernst Ruska, who invented it in 1933, but had to wait 53 years for his Nobel Prize in 1986, and he was lucky because he died only 2 years after he got the prize. So I like to joke that if you want to get a Nobel Prize and if you've done some very good work, you have to try and stay healthy and be alive. So the principle behind this is that if you have a three-dimensional object and you get an electron microscopy image, what the image is a two-dimensional projection of the object. So if you look at it from different angles, the projection will look different. And if you now take the Fourier transform of these projections, they will basically be equivalent to planes in what is called reciprocal space, or Fourier space. And if you collect all of the planes by sampling all the possible orientations, then you can inverse transform these and effectively reconstruct the object back. And that's really the principle of 3D EM. And this is from Aaron Klug, who got the Nobel Prize in 1982 for using this to do large molecular assemblies. Now, one way is to tilt the sample to get different orientations. But another way is that if you take your sample and flash cool it so that in a very thin layer of water, of aqueous solution, and if you flash cool it, then the water molecules are immobilized before they can form crystals of ice. And that's very important, because if you form ice, then you have enormous expansion and you essentially destroy everything. But if you flash cool it, then you get vitreous ice. So it's like a glass. It also doesn't create any background. You have a background, but it's more uniform. And these molecules are then trapped in many different orientations. And so when you get an electron micrograph, you see them in all different orientations. And then if you figure out which orientation gave rise to each of these things, then you can effectively reconstruct the object. And that's really how it worked. And big advance is that we now have better and faster detectors and better software to take advantage of the speed of these detectors, and better microscopes, so the particles don't move as much. And also, the detectors are fast enough that you can do motion correction. And as a result, you can now get high-resolution structures just using electron microscopy. And we applied that because all of us have mitochondria in our cells. These are organelles that our ancestral cells swallowed up 2 billion years ago. And now they're living sort of in a symbiotic relationship. And these organelles are where ATP, most of the ATP in the cell is produced. And they have their own ribosomes, which are descended from the ancient bacterial ribosome that they came from. But they have a very different structure. And so these two postdocs, Alexia Muntz and Alan Brown, solved the first structures of the mitochondrial ribosome from humans and large subunit from yeast. And then Nirupa Desai, who was a graduate student, worked initially with Alexia Muntz to solve the structure of the entire yeast ribosome. And you can see that here's the bacterial ribosome, this is the human ribosome, and this is the human mitochondrial ribosome, and yeast mitochondrial ribosome. And they look very, very different. All three ribosomes look very different, showing how different they are. Now, these structures would not have been possible using crystallography. In fact, many people, including we, have tried for many years. First of all, it's hard to purify mitochondrial ribosomes. They're not homogeneous, and they don't crystallize. But because of EM, you can now do many things that you couldn't do before. So I just want to conclude by saying, look, when I came, the last few days in Taiwan, I've talked to a number of people from Academia Sinica. One thing I realized is you've all been to top universities, okay? Cambridge and Berkeley and Stanford and all these places. Now, I didn't go to any of those places. I went to my local university in Baroda, India, and then I went to Ohio University, which is not a major research university. And then I had to shift from physics to biology, and then I had used this technique that was not very useful in biology, neutron scattering. So I had to, do all sorts of things. So I could never imagine that one day, I would have been in Sweden in December. And so the thing is, how could that have happened? So if you look at my life, it's sort of like a random walk, in science. Okay, and so one of the things I realized is I always try to keep my options open. So if I felt frustrated by physics, I could have left, and gone to some Wall Street-like job, or done something else, right? But if you wanted to stay in science, I thought, well, physics didn't quite work out, so maybe I'll do biology. Or if I learn neutron scattering, and that didn't turn out to be that useful. I thought, well, I'll go on sabbatical and learn crystallography. So I think if you keep your options open, it's useful. The other is I was never afraid to ask for help. So you just have to. And actually, when I went on sabbatical to the MRC, I found for the first time I was sitting in the same audience with all these Nobel laureates, and many of them would ask questions that I thought were stupid questions. Okay, they were like very basic questions after a talk, and I thought, wow, these guys are not embarrassed to ask these very trivial questions, and realized that you should never be ashamed to ask questions, because otherwise you'll always be ignorant, okay, if you don't ask. And so that's one lesson I learned. The other is many of the ideas that I got came out of random conversations, and one reason that LB is so successful is that we have a canteen. And in England, everybody takes off 20 minutes for morning coffee and then lunch and then afternoon tea. And they just go there. There's no appointment or anything. They just go to the canteen and they just sit next to whoever and they just start talking, And out of these conversations, sometimes new ideas come out. Somebody says, oh, I was at a conference and I heard this talk. You should look up this guy's work, So there are all sorts of little random things like that. But of course, you can't talk to people all the time because then you won't really think about your own work. And one thing I like to say is everybody has imposter syndrome, which is the feeling that you're not that smart and you fooled everybody. And one day they're going to find out that you're really kind of not that smart, And this is called imposter syndrome. And especially if you're a bit of an outsider, like me, came from India and didn't go to very good universities. Then you go to a top university and you think, oh, everybody here is probably a lot smarter than I am. And one day they're going to find out I'm an idiot. So I think that kind of thing is very common, and you should really just realize everybody has it. Even the people who go to top universities, deep down they're all a little insecure. That they don't know as much as other people. And then science doesn't just happen because of talent. Max Perutz, who founded the Institute, often mentioned the 4 Gs, which is you need Geld, Geschick, Geduld, and Glück, which are German words for money, skill, patience, and luck. So you need funding, otherwise, you can't do it. You still have to eat, even if you're a theorist, right? So, and then you need skill, which means you have to be trained properly. You have to have the right training and expertise to tackle it, or you have to acquire that skill. And then patience really has to do with not giving up too soon. There's a fine balance. If you just keep on doing something with no success, that's also not good. But if you give up at the first sign of difficulty, then you'll only do very trivial problems. You're not going to do anything that's really hard. And a lot of science depends on luck. Luck could mean you tried an idea, but for reasons you didn't quite understand, it worked out. Okay? And you only have to know the real reason later, why it worked out. You had no idea. Or you meet someone by accident at a meeting or colleague, and they give you some idea, which turns out to be work out. So that's also luck. So people forget the importance of luck. And when scientists become very successful, in their mind they revise history, and in their mind they say, oh, it was all a logical series of steps. Actually, it wasn't a logical series of steps. There were all kinds of little problems, and things worked out. So you have to realize that. So I want to conclude with this movie, just to show you how the excellent works. So this is the long molecule is mRNA, and this is a small subunit of the ribosome, and it binds in this little cleft. And this is the first tRNA that brings in the first amino acid. And those 3 other blobs basically help position the ribosome in the correct place to begin at the right point on the mRNA, starting with the first amino acid. And then what happens is that now the ribosome is ready to receive new tRNAs to bringing in the new amino acid. And we've already seen this process before. So you get, if delivering the tRNA, and then a peptide bond is formed. So the first and second amino acid are joined. Then the whole thing has to move. So the tRNA moves first with respect to the large subunit, and then with the help of another molecule, the tRNA and mRNA both move with respect to the small subunit. And the result is this site is now vacant, and a new group of 3 bases on the mRNA are exposed. And so the next tRNA can come in and deliver the next amino acid. And so this goes on at the rate of 20 per second, which is 5 times faster than what I'm showing you. And the tRNAs come in and out, and as the amino acids are added to the growing protein chain, the chain emerges from the back of the large subunit. And when it comes out, it can fold into a newly made protein, and then finally, there's a group of 3 bases with specified stop signal, and that's recognized by a protein which then cleaves off the newly made protein, which goes off and then does its thing. So you've made the protein. And then 2 factors come in and bind and split the ribosome apart, and the process gets started all over again. And while you've been listening to me, you have billions and billions of cells, and each of those cells has thousands of ribosomes, and they've all been churning out thousands of proteins during the time that we have been here. So thank you very much.